Lorraine is a leading voice in developing effective science-based strategies for managing invasive weeds in southern and eastern Africa, and I think there's nobody better to lead us through the next phase of this discussion. Lorraine, I'm going to hand over to you. Philemon, I would like to ask, if people are asking questions in the chat, if you can please start answering them in the chat, that would be really great. Because we might run a bit short on time to handle the questions at the Q&A at the end. Lorraine, thank you so much. We're handing over to you.
Thank you for the opportunity to talk to you about an introduction to biological control of invasive alien plants. And I'd just like to clarify, this is not native species that are bush encroachers, but exotic introduced plants that become invasive. It is a large topic. I will just take you briefly through an introduction to it. There are numerous more aspects to it, but we can discuss those later on. So one of the case studies that I'm going to use is Parthenium hysterophorus. I will allude to other species as well, but as Julie Kutsia referred to yesterday, when species are introduced from their native range into their invaded range, they are freed up from their natural enemies. And so Parthenium hysterophorus is native in the Central and South America, and it is invaded and is present in at least 50 countries. In its native range, it has a suite of natural enemies, hundreds of different insects and mites, pathogens that are associated with the plant, and these allow it to keep the plant in check. However, when a plant is then released from those natural enemies in its new country, it is able to have rampant spread and densification. As you can see, this is one of the conservation areas in South Africa, heavily invaded with this plant Parthenium. There are no natural enemies associated with it, or if there are, there may be a few guest native herbivores, but they really are not able to impact it severely. So because it has no natural suppression, it is now released from that. And in areas, as have been mentioned in Uganda, where there's disturbance, bare soil, high levels of overgrazing, or generally sort of land management that's not ideal, the invasive plants this creates like a perfect storm, and the invasive plants become really rampant. And they have no natural enemies to keep them in check in their introduced country. And we see impacts such as in agricultural production, in biodiversity conservation in our national parks, and around human residences, especially in rural communities, heavily invaded areas, in close contact with humans and animals. So biological control is the use of host-specific natural enemies, insects, pathogens, mites, and we refer to these as biological control agents, and they are introduced from the native range of the plant. And the idea is that they will reduce the plant in the invaded range to manageable levels and bring about long-term sustainable control. Now, this may be done with or without other control methods. So this is a graph showing the initial weed density at very high levels, the introduction of a biological control agent. As that biological control agent starts to increase in number and the populations build up over time, it then starts to suppress the target weed. And that target weed and the biological control population start to drop. They do fluctuate up and down, and ultimately over a period of years or even decades, they may reach a much lower level. And that is the target for biological control, as well as to increase the native vegetation that is desirable. So the aim of biological control is to reduce the invasive potential of reduce the invasive potential of the weed to a level where it is less problematic. And it is never, the aim of biological control is not to eradicate a plant. Generally, when these species are so invasive and at such high densities and extent, eradication is actually an impossibility anyway. So we should not be referring to them as eradicating unless it's at a very localized, very initial stage where chemical control may be a possibility to control a time. Not be referring to it as eradication, but management. So the advantages of biocontrol are that it's environmentally friendly, it's self-perpetuating, permanent. Once the biocontrol agents are established in the environment, they continue to build up in number, and it is cost-effective in the long run. The initial R&D phases are expensive, but in the long run, once the agents are in place, then it is cost effective, and it may also be integrated with other control methods, such as chemical control, not always ideal in the exact same areas, but mechanical control possibly, or competitive plant species, and other methods. Sometimes biological control is sufficient on its own, and sometimes it requires a combination of methods. So we can use biocontrol agents that attack the vegetative parts of the plant. They may feed on the leaves or the stems or the roots. And we could also, for an invasive plant, we could also affect the flower buds or the flowers and the seeds. And we can use those in combination. If a plant is deemed beneficial in some way, then we may just look at using reproductive attacking agents. So biocontrol in South Africa has had a long history. It's more than 100 years of work in this field. It started in 1913 with the introduction of Dactylopus salonicus onto drooping prickly pear. It then continued with sort of technology transfer projects from other countries, and there was a lot of work done on cacti and Lantana camara, and very successful projects reducing Opuntias using cochineal, various cochineal bugs, and to reduce them to more manageable levels. And some of them were really very impressive biocontrol programs and still are to this day. And yeah, work on Lantana camara. There's a lot of hybrids of this plant, so it has been a challenge. However, there are some very good biological control agents on that species. In the 1960s, 1990s, new weed projects were worked on that weren't worked on in other parts of the world, and Hakea was one of these. It was also very successful. In the water weed projects, there have been a lot of successes with floating aquatic species such as water hyacinth, water lettuce, Kariba weed, Sylvania, Pistia, and then also using pathogens like Euromycladium on Acacia saligna, and then 2 insect species on long-leafed wattle, and these have all been really effective. In South Africa, we were very fortunate that we then had a substantial amount of support from the Environmental Affairs branch of Working for Water program, and that really increased the capacity for work on weed biological control. And many biocontrol agents were then started to be released. And to date, we have released 136 biocontrol agents in South Africa. 92 of them established. 90 weed species have been targeted, and 66 have agents established on them. So we have a good long history. So there's some considerations for biocontrol. It's not a quick fix. The impacts may be subtle initially. So biocontrol agents may be released, but not very obvious, especially for the first few years. They increase with time, or they should do under suitable conditions, and they may be enhanced by increased numbers being released or over wider areas. And substantial suppression of the plant may need not just one silver bullet, as we refer to it, that is not that common, and because the plants have a suite of natural enemies, usually it requires a number of biocontrol agents to be introduced and provide cumulative stress to the plant. And then one of the key aspects of biological control is that we use insects or pathogens that are entirely can develop only on the target weed or have a very, very narrow host range, but mostly just monophagous. And we do not use species that have a broad host range. It is a rigorous discipline. We've got an excellent track record of safety. There are many steps in a biocontrol program. Pre-release stage involves work in the foreign exploration in the native range. We import then natural enemies into quarantine. We study them in quarantine. We try and establish breeding cultures and work on them further. And we undertake host specificity studies, which I will explain further. Once we are convinced, if we are convinced that the agent is host specific, then we will apply for permission to release that. That's from national governing authorities. And then if we receive permission to release that, we then move into a post-release phase where we start mass producing them, releasing them at pilot sites, and broadening their distribution to broaden their impact. We then monitor the establishment of the biocontrol agents, and we undertake post-release evaluation studies at selected sites. And ultimately, we may need to aim to integrate biological control into with other control methods. On occasion, we are able to some species are entirely controlled by biological control alone. So this is a long process. It takes sort of an average of maybe 4 years at least and especially longer for maybe novel projects. There may be some genetic work required to identify where the origin of the plant is and to ensure that we collect from climatically suitable areas, collect the biocontrol agents. So there is a lot of work around the pre-release stages and then obviously in the post-release stages. So we also need specialized facilities. We have temperature-controlled facilities such as glass houses and tunnels. And for some of the quarantine facilities are quite high security. They have certain specialized features, and there is usage is approved by national governments, by national government authority. And some of the countries that we've worked with, say in East Africa, they haven't had such high-tech facilities, but the principles of them remain the same to try and restrict the candidate biocontrol agents from escaping into the country before they are approved for release. We have various insect cages that we require, and the research is done within the quarantine, and once it's approved, it's done outside of the quarantine facilities. Another aspect is that skilled personnel are required in all stages, in the pre-release stage of the quarantine research, then in the mass rearing, and then in the field releases. And once we have released the biocontrol agents in the field, then post-release evaluation is really critical to ensure that that the biocontrol agents are doing what they need to do, that they're building up a number, and we assess aspects such as the growth and cover reductions of the weed, and at a more detailed scale, a lower scale level. And we also involve postgraduate students as well to assist with these studies. So there are a number of people involved at various stages of a biocontrol program. So host specificity testing, we go on to that in more detail. The aim of it is to determine to undertake a series of experiments to ensure that the natural enemy that we've introduced into quarantine will only survive on the target weed. And it can't survive on any other plants that we don't want it to survive on, any economically important plants or anything else. and that we can prove that it's safe for release into the introduced country to control the weed. So we have, this is quite an outdated theory, but it's still, there are more modern theories that as well that have been introduced in terms of host range. But the principle of it is that we would test, so if our target weed was Parthenium, it's in the tribe Helianthiae, so we would test closely related plants that are closely related to the plant within that tribe. And then as we work our way out in degree of relatedness, so less related plants, we will still test a selection of those. For example, in the Asteraceae, there's something like 2,000 odd species, so it would be impossible to test all of them. But if we are testing the closely related plants and there's no development on those, then we are quite secure that there will be no development on something that is totally unrelated in a different family. Okay, so then the types of host specificity tests that we undertake are no-choice tests. Those would be very conservative tests where we would put the insects, a set of insects into each cage, and they would be basically, they would have to survive or die on those plants. So it's really conservative. We're forcing them, and we would evaluate their survival and their egg laying, their feeding, and what happens to them during a period of time. And that period of time could be a week, it may be 2 weeks, maybe longer, but generally there's those sort of time frames. If we those plants that we don't want them to attack, we would then take them into a paired choice test where we would offer them the choice between the target weed and the test plant, and see again what sort of outcome there would be, because this is a very artificial environment in a glass house, in a insect cage. It's a very confined space, and so they may behave differently to what they would do in the natural environment. Should we still see some results in there, we could also take them into much larger walk-in cages where we would expose them to multiple test plants as well as the target weed. So for an example, in this setup, we would have 5 pairs of weevils, and we would expose them to the single target plant on the top here. Up here, this is our test plant. This is our control plant, Parthenium. So this would be in a separate cage, and we would expose them. And at the end of this, we would count the number of eggs that are laid on the flowers. These are, I don't want to focus on much of the detail, but this is sort of the table of the first round of testing of no-choice tests. What I'd just like to show you would be, these are the number of eggs laid relative to the control. So your control being your test, your weed, Parthenium. And you can see there, although there may have been a few eggs laid on them, they were very low in number relative to the control. However, they were still a concern. There were a lot of zeros where there was no response of the weevil on it, and it was really the ones that were all within this same tribe. So then and as you can see, these are lesser related species, and there was no opposition except for one. So we then took them into choice tests and paired choice tests or multiple choice tests, where we exposed a large number of weevils into a walk-in cage. We staggered the plants, we rotated them every few days so there's no light effects, and counted the eggs after that. So then we still had a few eggs laid on some of the species, so we then did larval development tests where we put eggs onto the plants, onto the non-target plants, and we assessed their survival and development. We then ran a risk analysis, and that can be used in cases where we may have had feeding or opposition on some of the non-target plants in the earlier stages. And what it allows us to do is to actually quantify and give a number to the outcome that we see. And that is a much easier assessment to make for decision makers. It gives a number, so it would maybe be like 1 would be on a figure of 1 for the target weed, and 0.1 times 10 to the minus 9 or something like that. So a tiny, tiny, tiny figure. And it really gives a relative comparison of the risk associated with that. And we use native range records and host specificity results from other countries. That's always, if there's results from other countries, that's always useful. And in these cases, it was decided that the agents were sufficiently specific and they were approved for release. So the key points are that host specificity research is very rigorous. We have an excellent track record globally even, and particularly in South Africa. More than 480 biocontrol agents have been released on 187 target weed species, and there's been no significant unpredicted non-target damage in over 100 years. So in some cases in the early days, there has been a spillover onto some species, but in South Africa, that has not happened at all, and we have an excellent track record. The agents released are host specific, so we don't expect unpredictable non-target effects in the field. We've spent a lot of time ensuring that they are not going to cause problems. And I know the burning question on everyone's mind is always, what happens when the weed has declined or diminished to a low level? Will the biocontrol agents not become a problem themselves? And the answer is no, because they are closely evolved with the plants. So as the target weeds decline in abundance, so does the biocontrol agent. And again, if the target weed increases, we would expect the biocontrol agent to follow suit. Sometimes there's a bit of a lag phase, but that is the curve that it does follow. So we don't expect them, especially for classical biocontrol using introduced insects and pathogens and mites, we don't expect unpredicted non-target effects. As I mentioned, we may get false positives during the confines of working in a quarantine environment, and those are not likely to occur in the field. And also, we don't release every agent that we bring into quarantine. If the researchers are not confident from the outcome of the testing that the agents are sufficiently host specific, they will not be even applied for release. So there have been a number of agents that have been rejected. The R&D for biocontrol is a lengthy time, and establishing them is lengthy, but the outcomes and the long-term benefits are really beneficial. And yeah, of course, because we're working with imported natural enemies, it is really critical that we have access to new natural enemies and really robust relationships with and collaborations with native range collaborators in institutions in the native range. So in, say, Central and South America or Australia, Europe, it's really critical, those relationships, for the success. So once we have an approved biocontrol agent, we can mass rear it. This is a large scale production, and yeah, it's needed to establish the agents as widely as possible. It's really critical for the success of weed biocontrol programs. There's not much point in putting a lot of time and effort into the developing a biocontrol agent if the resources are not provided to rear it and distribute it as widely as possible and to make full use of its benefits. It's basically a production line where quantity and quality are really important. It may require sustained efforts. It depends on the plant and on the biocontrol agent. Sometimes multiple releases are needed to achieve establishment. It depends on how fast some agents can establish from one release, and others may need multiple releases, or we may want to use multiple releases to improve the levels of control that are achieved. So some of the elements for mass rearing that are important are obviously plant quality and quantity. That's our starting blocks. We really need to ensure that they are appropriate for the biocontrol agents that we are trying to produce, whether they are insects or pathogens. Obviously, appropriate facilities. These are daily activities. It includes Christmas Day and New Year's Day. So there needs to be dedicated personnel that are always assigned to these activities. There needs to be rapid troubleshooting to ensure that there's good outputs from these facilities. The scale of production is important, and obviously good record keeping and research that then feeds back into improving mass rearing programs as well. So all of these stages are important, and they influence the outcome and the success. So plant production, one would need continuous plant production, even if it's during a dry season or a winter season. One would still need to be ready and maintain those cultures through those periods. And the structure of the plant also needs to be suitable for, if it's a leaf feeder, one would need large leafy plants. If it's an insect that's galling the shoot tips, one would need a bushy plant with lots of really robust shoot tips for it to lay eggs on and to feed on. Obviously, the number's important. There's fertilizer, the whole plant production side of things, pests and diseases, scouting and treatment of those. And then there's the insect production side of things, ensuring that there's adequate space and resources and that there's a continual supply of fresh plants for feeding, mating, oviposition, and then development of the juvenile stages and until the next generation of adults ecloses and is then replaced into the breeding cages or released from these facilities. So there's daily activities, record keeping on large scale. The mass rearing is done ideally under temperature controlled environments. However, we have worked in facilities where there's just sort of external and there's just ambient temperatures. Obviously, the outputs are less than in being able to work in a temperature controlled environment. But it is possible to do it on a more basic scale. And then both the plants and insects or pathogen, insects certainly, they have a seasonality, so they may decline during the drier season or the wetter winters or during basically when temperatures drop. And we need skilled personnel, constant water and electricity, and then the scale of production is influenced by the resources and capacity. Once one's able to do the field releases, there are elements such as transit conditions that are important to select climatically suitable sites. And obviously things like heat, extreme heat, drought, flooding events, those all influence establishment. It's helpful to have pre-release scouting of sites to identify something suitable. And there's characteristics such as the temperature, rainfall, elevation, slope, microclimates that can all affect establishment of biocontrol agents. Plant quality is important. It needs to be an extensive infestation to ensure that there's enough food and that the agents are able to ultimately move off. They may breed in the area for quite some time before they are able to disperse, but some disperse quite quickly. And then, obviously engagement with landowners and things like we do research around the size of releases and stage of releases and trying to determine which are the most robust stages to release and what number and how often. And so there's research components around those elements. And obviously all the monitoring and evaluation after those releases as well to ensure that that the agents are establishing and ultimately to evaluate what kind of impact they're having. So with the field site selection criteria, we may also use tools such as, laboratory thermal physiology research and feed that into climatic modeling. And this would provide outcomes that we could see where, okay, so like the black dots would be where the weed is, and then the darker the red blocks would be higher areas that are more predicted to be more climatically suitable for the agent. And we do use these models. They can improve the outcomes of establishment. They're not fail-safe because there are multiple factors that influence biocontrol agents once they're released in the field, but they are helpful in directing releases. For Parthenium in South Africa, there was actually a national strategy that was developed and an implementation plan, and this was for a period of time, and that was then updated after 5 years or so, and that directed efforts. And so the country was basically divided into zones where we would use different control methods. So in the purple sort of areas, where Parthenium was not present, this would just be early detection, rapid response, and the red areas would be use of chemical control, and those would be very small populations, or populations of the weed that we would think that could be brought under control by using chemical means. In the orange areas, those are areas where the weed is much more dense, much more extensive. Chemical control is also used in those areas, and biological control could start to be implemented in those zones as well. In the asset protection zones, those were almost sort of really undesirable and really not feasible to be spraying herbicides to the extent of the weed invasion there. And this is where biological control is also really comes into its own, where there are areas that it's not suitable to be using. Chemical control could be used along roadsides, clinics, schools, key asset areas. So biological control is then designated for certain areas, and the chemical control, the early detection, rapid response programs can operate in different areas. Obviously, in the areas where they are both operating, there needs to be cooperation and a lot of collaboration so that biological control agents are not released, and then a chemical control program comes along and just sprays the plant, and the insects or the pathogens are damaged. So there's communication needed there. So biological control is not a suitable method for weed control at all sites, but it is the most appropriate where alternative methods are just not physically or economically possible, or where it would cause unacceptable damage. So, and those are many areas certainly in South Africa and many countries where people just don't have the resources, sufficient resources, or the weed is so extensive that means methods such as biological control can be implemented. And it's not just biological control. We'd also be possibly looking at competitive plants or some sort of physical means. Someone mentioned using fire for chromalina in certain areas, but biological control is also really effective in some areas as well. So an integrated plan is always needed. In terms of field site selection criteria, as I've mentioned, some established readily from a single release, and others require numerous intensive efforts, and we refer to those as augmentative biocontrol. The agents are pre-programmed. They're inherent, whether they disperse rapidly or slowly. Some barely disperse more than 500 meters in a year or 2 years, and some will disperse 50 kilometers or even beyond that in that same time frame. Some of the biocontrol agents have dispersed through 400 kilometers across oceans, up into East Africa even. So it really depends on the biocontrol agent. And a key thing is that we need secure sites. Obviously, the type of plant, whether it's an annual or perennial, affects the requirements and the individual requirements of the agent. Maybe they only feed on a certain stage of the plant when it's really young or when it's more mature. And biotic factors, soil nutrients, moisture, those aspects also have an influence on plant quality. The biocontrol agents may have certain preferences. Some prefer shade or open areas, young, mature plants, and then they also need to be close to other infestations so that they are able to disperse. There may be host plant biotypes, or races or hybrids, such as with lantana. So those also need to be taken into consideration. We request at least 2 years of no disturbance to a release site, and if possible longer, to ensure that the biocontrol agents have time to establish. Yeah, there's things like maybe if one releases along the edge of commercial crops, they may be affected by spray drift of pesticides. This is not always the case. Biocontrol agents are able to establish in some of those areas. And yeah, where there's homesteads, they may be cleared directly around. There may be a high level of mechanical removal of the plant. So one would tend to release the biocontrol agents a little further away. And then once the biocontrol agent is very well established and very abundant in certain areas, they can be then designated as biocontrol reserves, and those can be used for harvesting and redistribution of the agent. Obviously, we're talking about several years or maybe even a decade, probably 5 years at least, for the agents to establish to a really good, abundant level where they can then be harvested and redistributed to other areas. So the biocontrol agents always need to be released at many different release sites under different conditions and in different regions firstly to establish them as widely as possible, and then also to try and establish them in different conditions, because they may establish well in certain sites and not well in others. And those are always specific to each biocontrol agent or what they require. So an understanding of the requirements per agent is really important. And the site suitability in the plants change with time as plants mature and there's succession of other vegetation. So it's always developing. With Parthenium, this was actually a major problem in central Queensland in the 1980s. In Australia, really, really dense infestations like we see in a lot of Africa. And they had a very, very successful biological control program. They introduced 11 natural enemies. They also used chemical control along the roadsides, but the long-term control was also achieved with a really large suite of natural enemies that targeted the different parts of the plant. And this was reduced from being the major weed, the #1 weed in rangelands, to still present. It's still present in Queensland, and it is problematic in small areas, but it is nothing like the extent that it was in those years. The biological control obviously has taken time to build up in number and impact, but even in the 1990s or so, the benefit to cost ratio was about 7-1. So for every dollar spent in costs, benefits of at least $7, and that was in the 1990s or so. And it's really just with time, those benefits increase. This is a slide of introduction, the timeline of introductions of the biocontrol agents, and some of them, sort of the bigger the error, the longer the time that it was actually before they were evident and common and what from the time from introduction. So some had really rapid establishment and spread, and others took maybe 10 years even before they became observed in any great number. So it just depends on the different biological control agents. In South Africa, we've got 5 different, we've got 2 rust fungi and 3 insect agents that are present in the country. And we're also working on others, on a root crown boring moth, because we need to increase the level of damage that is happening and the extent of it. But after, say, about 5-10 years, we are seeing agents spread sort of, we're detecting them, say, 3 to 5 kilometers from where they've been released. We're seeing damage to the interior of stems. This is a seed feeder. and it's able to cause abortion of the seeds and within the same flower head. And those numbers the weevil is becoming more abundant and is dispersing, and we're seeing them becoming more common and more abundant. And the level of control, although it was measured at about 30% reduction of the seed output within the first few years, we would expect that to increase well beyond that in the decades to come. We also, certainly for Parthenium, as well as many of our other weeds, we collaborate internationally. But for Parthenium, there was a USAID funded project, and it allowed for the introduction. We did introduced the leaf feeding beetle and the stem boring weevil into Ethiopia. There was a lot of mass rearing done there that both of the agents have established. They were also introduced into Uganda. I am fairly certain that they would have established, or at least one of them will have established. There were also shipments sent to Kenya and to Tanzania as well. But those would need further development, more mass rearing there, and more interventions as well. So we've trained a number of different international scientists at our labs, and our colleagues at the Center for Biocontrol. They also have a number of foreign scientists that they interact with, either in country or within South Africa, with training programs. They offer an excellent weed biological control foundation introductory course. And yeah, we've also as part of the East Africa project for Parthenium, we also helped them to set up a quarantine facility from scratch, introduce the agents, taught them all about the host specificity testing. They ran their own host specificity tests, and they were then they set up a mass rearing program and distributed them widely throughout the country. And these programs really need a lot of national support once a foreign agency has removed the funding. So it's always critical to have national funding as well. So about in 2017, there were about 76,000 of the leaf feeding beetle released in Uganda, and about 10,000 odd of the stem boring weevil. So I would be very interested to engage with people there further. As I say, I suspect that at least one of them has established in multiple sites. But we could discuss that further. So yeah, after field releasing the biocontrol agents, we need to assess the establishment. We need to look at changes in the abundance of the agent. We need to monitor their spread. This may take several years or longer to become for them to become common, and they often can only be determined. Their true potential can only be determined many years after it becomes well established. And it's really important to undertake post-release studies to quantify, there's no the monitoring and the evaluation is really important to look at what impact the agents have on the plants. And these may be variable from year to year as well, and there may be hidden effects that are not obvious to the untrained eye, but they may be an internal feeder and they're still having an impact on the plant. Then measuring success, it's always important to measure success. It can be quite tricky at a larger scale. At a plant level, it's relatively easy. We may need to use chemical exclusion trials or long-term transects. And yes, people sometimes say that a biocontrol agent, a target weed is still very problematic, but they have forgotten the dense extent of them decades before. So in South Africa, we've got a rating system for our biological control programs and to categorize the outcome of the weed biocontrol. This was originally on a fairly simple system with complete or negligible or substantial control, and we realized over the years that we needed a more nuanced approach to this. So in more recent years, a system has been developed to categorize them based on density and biomass and area and the rate of spread. And so they're given A's or B's or B pluses or C minuses, and the combination of those allow us to have a more nuanced evaluation of these biocontrol programs. And if we look at those, complete control is in about 33% of our programs, and most favorable is in about another 10% of those. And then there's sort of there's varying levels of control achieved in the other programs. So there is some level of control in most of the weed biocontrol programs. Some of them are outstanding, and we have no need for any other interventions. So we can measure success in different ways. We can reduce the plant density, or we want to know whether the plant density and biomass has reduced on a population landscape level. And we have very successful programs such as the Red Hispania, and this was a synergistic effect of using 3 biocontrol agents on pods and stems, and those really did an outstanding job of reducing the biocontrol agents to a much the target weed to a much lower level where it's no longer problematic. The red water fern had a much quicker turnaround time within a matter of months. This tiny little weevil that feeds on the fronds was able to destroy the red water fern, and this is a really really was quite an outstanding program. It's not often that we see such quick turnaround times of the impacts of the biocontrol agents. Stricter, many of our cacti projects are very successful using different cochineals. It's important to have the right biotype of the cochineal, and you can see the decimation of using just the cochineal, and this is in one of our national parks, and really successful programs. Again, the Acacia sideligna also with the fungus, and there's an exceptionally good mite that affects certain of the hybrids of the lantana, and then black wattle, which is a useful is used in some areas in our country, but it's incredibly problematic and has a 50-year seed bank that's have been difficult to control, and it's there's a very effective galling midge that substantially reduces the seed bank to sort of just a few seeds per square meter in some of the areas where it's climatically suitable for them. And but we can use, was a situation where also we can attack reproductive tissues of a plant that has beneficial uses as well. Then there's, of course, all the economics associated with measuring success, and Van Wilgen and many of his colleagues have done some outstanding work of trying to assess the economic benefits of the existing biocontrol programs. And some of these have reduced invasive plant management costs by 20%, with the potential to reduce that even further, and that was in the 1990s. Of course, with time, those benefits just increase. And yeah, so we have really, we're getting benefit cost ratios of 1:8 and up to 1 to almost 4,000. So really, really outstanding benefits from the investments made. And obviously another way of measuring success is to measure the recovery of natural vegetation, and various other measures of biodiversity that can be assessed. Ecosystem services, increased agricultural output, increased water supply, and then socioeconomic studies. So those are all various studies that can measure success of a biocontrol program. Just to draw your attention, there are many resources. Obviously, there are numerous published papers in scientific peer-reviewed journals, but there's also a world catalogue of target plants and the associated biological control agents, and that's a global compendium that's updated on a regular basis. There's the local journal, entomological journal, and decadal reviews are published from South Africa every 10 years on biocontrol programs. So there's 3 of those already. And the status of biological invasions in South Africa is a status report that's compiled every few years. And it obviously also incorporates biological control and many other management interventions. There is an international organization for biological control that has an umbrella body over different regional sections. And there's an Africa tropical regional section. There is an international symposium on biological control of weeds that's held every 4 years. The next one's in 2026. That is specifically about weed biocontrol. Is an excellent conference. In terms of Parthenium itself, there's a book that was developed recently and published in 2019, and that covers all sorts of elements about Parthenium weeds, biology, ecology, and management. Has multi-authored chapters from around the world, and more recently, as an output from the East African project, the biocontrol program on Parthenium in Ethiopia was written. The efforts were written up into a book, so that's also a very handy resource for countries that are stepping out into a program of that. And then finally, I'd just like to say that biocontrol it's a really essential tool for sustainable, cost-effective management of extensive stands of invasive alien plants. As I mentioned earlier, if there are very tiny weed populations, one may use chemical control or something to really stop the weed in its tracks before it spread really further. But for areas where there's resource poor areas or where the weed is in such great extent, then biological control is really it's an essential tool. There's an excellent track record of safety, and there are very many successes, all published. And yeah, it really is a very rigorous science. Sometimes a suite of introduced natural enemies it's often necessary. Not often that you'll get a single biocontrol agent being able to bring it under sufficient control. In some cases, in some of the water weeds or some of the cacti, this is the case, but not in all target weeds. And it's important to maintain realistic expectations of outcomes and timeframes. And patience is really an important thing. Some of the biocontrol agents may be released and they've thought to have not established or have no impact. And a decade down the line, they suddenly become extremely abundant or they become common, and their impact seems to be more detectable. And I think possibly there's frustrations with some of the programs that have introduced agents, but the expectations were beyond what was achievable at that point. But with further implementation, these could be improved. The biocontrol activities do require long-term support from national governments, and there are numerous invasive species in common in Africa with biocontrol options and expertise available in South Africa. I mean, the waterweeds, the cacti, lantana, tithonia, tacoma, chromalina, parthenium, campyloclinium, cardispermum, xanthium. There are numerous biocontrol programs that we are working on that would be of benefit to our African colleagues. And then, the impacts of biocontrol increase with time, and the returns in the investments increase with time. So I just like to acknowledge our past and current funders and various biocontrol colleagues for some of the images. Thank you.
Thank you, Julie, Lorraine, for this comprehensive and in-depth overview of biocontrol. Certainly fascinating. Lorraine, there's been a lot of discussion in the chat while you were talking, and a lot of it has been answered by Julie quite comprehensively. Julie has been writing the concise guidebook to biocontrol in the chat, so you guys can just copy and paste there. But for those of us who are not so good at multitasking and following multiple tech platforms at the same time. I would like to ask some of them for you as well, so you can answer them in video format. So there's, I think, an overarching question that was started by Lawrence. What happens to the introduced pathogen when the targeted plant species gradually reduce? Won't the pathogen then become invasive also?
Okay, so no, that is key to the crux of biological control. So if you, I'm not sure if you remember, I know there was a lot of content in the presentation, but the graph at the beginning where there was the biological control and the agent and the weed track each other. So the biocontrol agent increases as the weed increases, and as the weed decreases, the biocontrol agent follows that. These are closely, certainly with the insects and pathogens, they can be extremely host specific. I mean, some of them won't even feed and develop on another, like if there's lantana and there's different hybrids of it, they will only like a certain color variety. They require a number of sensory cues to be attracted to a plant and to feed on the plant and to mate and to lay eggs and to be able to develop and sustain populations. So the biological control agents, both insects and pathogens that are used in weed biological control, are completely host specific. We do not release biocontrol agents that are going to become problematic on other plants. So they will decline with the weed. They will not become, and we test these, in our host specificity tests in quarantine. We test them in a number of different scenarios as to how they will react to being just starved on a plant, on a plant that's not their target host. And yeah, so it's a very common concern, but it's because we're not using sort of species with broad host range. So we're not using a prolificous agent that's going to eat a number or lay eggs on a number of different types of plants in different families or different tribes, and we're using they're very closely honed to the target weed. They cannot survive basically without the weed.
A number of people raised concern then for the survival of the biocontrol agent, and Bosco asked, are there also studies on the survival of the biocontrol agent in the target areas, e.g. risk of predation by birds, etc.?
Yeah, so that certainly is a concern. Local predators and in time, even parasitoids, if they are closely related, say there's a weevil and there's a closely related native weevil, it may have parasitoids that could in time maybe move on to the introduced biocontrol agent. So what we overcome those by introducing large numbers of the biological control agents over a period of time and in multiple different areas. So there are ways around overcoming those concerns. I mean, there can be a concern, and we would maybe physically put them, so we wouldn't, if we release them, we wouldn't put them onto the soil surface where ants may immediately carry them off. We would, and we wouldn't put them on the top of a plant where the birds would maybe be predating on them immediately. So we would put them halfway down the plant so they've got a bit of shelter as well, and then they will move off. So they are, I mean, these are natural systems as well. They are used to having predation and parasitism, and it's even higher in the native range. They're actually freed from their predators and parasitoids more in an introduced range, so they should be able to establish even higher populations in the introduced range until perhaps some of the other predation effects might or parasitism may become an issue. It can be an issue with some biocontrol agents, but not all.
Thanks, Lorraine. I was going to turn to David's question in the chat next. So great, he's got his hand up, so you can ask it live. David, over to you.
Okay, hi, yeah. So just a quick question. You're introducing all these hundreds of thousands of new biocontrol agents every year. Have you tested how that impacts the predation of local animals? So normally, let's say a bird or something will eat one type of insect. Now there are hundreds of thousands or millions of these biocontrol agents. Has that been studied? And what is the impact of the local ecosystem of introducing so many foreign or exotic animals year after year? Have any studies ever been done? Like, just for an example, water hyacinth weevil, millions of them get released. Are you not now pushing your things that used to predate mosquitoes to predate weevils? So that's a drastic example, but has something like that ever been studied?
There are, yeah, I think there are some studies that have looked at that, but I mean, generally, I mean, it's kind of we wish that our biocontrol agents would get to enormous numbers that they would become such a problem. But generally, we're more content with 500 or 1,000 or 5,000 or 10,000, and these are sort of localized areas. So on a landscape scale, I don't think they would be having an enormous impact on a species level at a landscape level. I would say, yeah, I mean, there've been a few species. There was a defoliating caterpillar that was released in but over a period of many, many years, there would maybe have been 1 million released. Some of the water hyacinth, megamelis, are being released in really high numbers, but I mean, they are feeding on those target plants, and once the plants have dropped down in the system, have declined and reduced, those insect populations will decline with them, which is why they have to keep inundatively releasing them as well as all the eutrophication and being flooded and washed downstream in it.
Thanks, Lorraine. Gustav, you're welcome to unmute.
Thanks, Johan. Quick question. How does the biological control, if you're using an integrated approach, get affected? I think you partially answered this by herbicide and by fire and by other, let's say, browsing and all of those methods. Or, I think I caught some of it, that obviously species specific, but yeah, just like in general, how do you see that? Thanks.
So we have done some trials, in our glass houses where we've exposed biocontrol agents to plants that have been sprayed with herbicide, and it definitely affects their mortality, much higher mortality, less egg laying, less feeding. So it's not complementary to have those two methods at the exact same sites at the same time. But there are, like in the water weed biocontrol programs, sublethal herbicide levels of herbicides have also been investigated, and they may strip spray with chemical herbicides on some of the river systems, but leaving a reservoir for the biological control agents, so that they can then build up with time as the weed populations build up again. So it depends. I mean, some of the biocontrol agents may have a soil dwelling stage. If the fires are quite quick that burn through there and don't reach very deep in the soil, perhaps they would survive. I mean, they're also coming from natural systems where there would be fire and other mechanical interventions being used in their native range as well. But obviously we're trying to sort of protect them as much as possible in the initial stages of establishment. The first 5 to 10 years at least, where they're trying to get a foothold in the environment. So it is dependent on the different biocontrol agents and the different interventions that are used. There's some studies, but more of sort of being undertaken to try and integrate biological control into management methods. I don't think they're ideal across the board yet, but yeah, there is research being done on those elements.
Thanks, Lorraine. Joseph, you're welcome to unmute and ask your question.
Thank you so much, colleagues. Good morning, everyone. This is Flavian Turanzomwe, species specialist based in Murchison Forest Conservation Area. I have one question to Lorraine on biological control. It appears as if it requires extensive research and tests, because like before release, there must be tests either on host specificity. You must conduct studies on environmental impacts, and again, there must be approvals. Can't this method of biological control become more expensive and slow to implement? That's my question.
So it can be. I mean, it's obviously not cheap, but nor is developing a chemical herbicide or something. Then R&D of any process involves some sort of investment. The thing with biocontrol is that once the biocontrol agents are established in the environment, that they should be able to. The idea is for them to have self-sustaining populations. So it's not a continuous investment for the next 50 years or something like that, but you need sufficient to get it off the ground. The other element is, firstly, there may be a number of the biocontrol agents may already be present in Uganda. I'm sure there's a number of agents on lantana, possibly chromalina. There's some very good. There's a couple of good biocontrol agents, a galling fly and a leaf-feeding moth, and they disperse themselves. They don't recognize country borders. And then there is also the willingness of each country to accept if, for example, there was a program, there was a long-standing program from 2005 to 2022 on biological control of Parthenium in Ethiopia, and those agents have established there. They will move across borders. Some of them have in Southern Asia have moved across countries from India into Pakistan into Nepal. So some of them are able to disperse beyond that. So it's not always that the entire investment has to be spent in a program. So if a country is willing to accept the host range records, and I really think it's something we need to move towards as a regional approach, that there's acceptance of the host specificity testing from other countries, and it particularly from within regions as well, and that because the countries that are introducing biocontrol agents, those agents may move in time across to the other countries. I mean, that's ultimately what we would like to see, that there's a self-sufficient, self-sustaining spread of them as well. So it's not. So some of them can be they're sort of piggyback projects that are always cheaper. They don't have to be. The technology transfer doesn't have to be done from the initial ground up necessarily. So it depends on the country and their policies and their willingness to do it. But I would also start with surveys and just seeing what is already present on those on the invasive plants in each country as well. And then Kenya, Uganda, Ethiopia having some sort of a regional committee or something that they then discuss and have agreements on acceptance of host range testing, and in one country is then acceptable to another, and that introductions can be done, because the reality is if one country is putting a large amount of investment into it, but their neighboring countries are not, there's a continuous seed spread into the country, and so it just sort of slows down the program. We can really enhance the biocontrol efforts that have been done.
Thanks, Lorraine. That's really a very important element. Then I would like to turn to Graham for the last question, and then invite all of you, if you have more questions, please post them in the chat, and Lorraine can continue with answering there. We unfortunately are running late in the program at the moment, but it's because of your wonderful participation. Graham, over to you.
If I may, good morning, everyone. If I may make one comment, Lorraine, you mentioned that using sublethal doses of herbicide to give the insects a chance. That is a highly dangerous technique to use. Sublethal doses on annual and biannual plants tends to lead to herbicide resistance. If you're going to spray to limit the plant strength, spray swaths and leave some untreated. Do not spray sublethal dose. It's illegal and it's dangerous. Thank you.
Thanks, Graham. Yeah, I believe it has been the legalities have changed, I think. Yeah.
Okay. Thank you so much. Thank you for the caution on that. Lorraine, thank you very much for fielding the questions and the really fantastic overview.